My pleasure to introduce Dave Odde, who I've known for quite a while. He did his undergraduate work at University of Minnesota, went to Rutgers for his master's and PhD, and then came back to University of Minnesota, I think in 1988.
99. Yeah, I graduated in 1988.
Oh yeah, that's right.
Went back there in 99.
Back in 99 for as an assistant professor and rose to the ranks, and he's an expert in modeling the mechanics of migration and then validating those models in real measurements. So awesome. Thanks for joining us and look forward to your talk.
Yeah, thank you, Jan. And thank you, Forrest and Francisca, for inviting me to participate today. And we've heard earlier about the invasive nature of glioblastoma, and that's a major focus for my group is how do they do it? How do these cells crawl through the brain tissue in order to spread the disease and the mechanics of that? And if I have time, I'll tell you about the mechanics of immune cell migration as well. I decided to squeeze in the middle between those two stories a crazy pitch for a clinical trial just to see where it lands. If you shoot me down fast enough, I'll just go right on to the immune cell mechanics. So it's a talk that has three halves to it. So I decided late to add one half in the middle, which was always a mistake, but we'll see. Okay, so I have a wonderful group at Minnesota on the left here, and I've been really mentored and encouraged by two key people at the bottom here. I'll specifically mention Steve Rosenfeld, who's at Mayo Clinic, Jacksonville. And Steve's the one who's told me I might have something to contribute to the brain tumor space. He's a neuro-oncologist and a molecular biophysicist, and he saw what we were doing with modeling neuron growth and mechanics and thought that could be applicable to brain tumor invasion and mechanics. So Steve's been encouraging me for the last 10, 15 years to try to apply our imaging and modeling to brain cancer. And being at Minnesota has introduced me to Dave Larga-Espada in the lower left, who's the associate director of our cancer center and outstanding cancer geneticist and expert on brain tumors and other tumors as well. And of course, benefited from the generous support of the people of the state of Minnesota and the people of the United States who have supported us continuously and generously through the National Cancer Institute for many years now. So thanks to all of you. And we heard earlier about the invasive nature of glioblastoma, and you can see it in this sequence for this one patient, which was one of Steve's patients, who presented with the disease on the left and underwent surgery, radiation, chemotherapy, and as Jan mentioned earlier, almost inevitable recurrence, which you see in the middle image, only 3.5 months later. And that recurrence in this case was very close to the original lesion. And over the next 2 months, despite entering a phase 2 clinical trial for a new immunotherapy, this individual progressed even further to the point where the whole left hemisphere is basically taken over. And 2 weeks after this last scan, perished from the disease. So 6 months from being like you and me, sitting here feeling pretty good, to being deceased from this terrible disease. So I thought I'd just mention the invasive nature here specifically, and being able to disrupt the cell migration might be a therapeutic opportunity to limit the disease's spread, keep it more local and confined and compact tumor, so that local therapies like surgery might be more effective at limiting the tumor mass and invasion, and also focused radiation might be more effective as well, as long as we can keep the tumor more local and keep it from spreading further into the brain. So that's motivated us for a while now to try to figure out what are the mechanics of how they do that, and then does that give us clues on how to disable it. And at the same time, we're interested not only in the glioma cell migration, we're interested in the immune cell response and how immune cells navigate through the brain in order to effectively, in the case of anti-tumoral cells, like cytotoxic CD8 positive T cells, find the cancer cells, and then when they get there, kill them. So it's a tough finding problem because the diffuse nature of the glioma means that there's big gaps between one cancer cell and another cancer cell. And so you're asking T cells to go from one to another. It takes time to find the next cancer cell. So maybe the time to kill it maybe isn't that long, but the time to find the next one can take up a lot of time. Meanwhile, time is ticking by, and the cancer cells keep growing, and they keep moving, and they keep dividing, and so they can't afford to spend so much time looking for cancer cells. And maybe they're not all that well equipped to do it. So we want to understand the mechanics of those, particularly for cell-based therapies, to make sure that those cell therapies that we're introducing actually are capable of navigating through the brain, which they have to do in order to go from one cancer cell to the next in order to keep serial killing. So we're interested in both of these aspects of migration, both the cancer cells and the immune cells. I'll focus first on the cancer cells, and just showing a histology of, again, the diffuse infiltration. And the surgeon colleagues that I have say that they're comfortable resecting up to this, if they can find it exactly, this kind of margin here, where there's a tumor mass that's clear that they can identify, beyond which is tissue which has clearly been invaded by cancer cells, but is still more or less functioning neural tissue. So they risk, by resecting that functioning neural tissue, they risk severe debilitation or worse for their patients, and they're not willing to take that risk. And therefore, they view this surgery as largely a palliative treatment that is worth doing because it improves quality of life, because it debulks the tumor. But by itself, and even with other combinations and standard of care, do not cure people. So that's the problem, is these cells are being left behind, and they continue to keep invading and progressing. And they're kind of sparse as you get out to the edges, making it hard for the immune cells to find the last few that are out there, because there's such great distances between them. So if we could keep them from doing that, we might be able to keep it more compact tumor. So once immune cells do get in there, they just keep killing locally because they're all right there. So here's what it looks like more in a live, using confocal microscopy imaging of, here we've induced a tumor using a method that Dave Largaspada's lab developed, and fluorescently tag the glioma cells with GFP. And you can see individual green labeled glioma cells invading into the brain parenchyma to the upper right and away from the tumor mass in the lower left. And they go at different speeds, which we can measure. So we can start to see the speeds at which cells are actually migrating in brain tissue in these ex vivo brain slice assays. And that gives us a handle to start to look at what the genetics and the drugs are that we could explore to understand the mechanisms by which they're doing that. And you can see this in this, not just in brain tissue, but in brain-like tissue, now using human iPS cell-derived brain organoids as the platform, and interfacing that with green fluorescently tagged glioblastoma cells that can attach to that organoid, ingress into it, and migrate through it, and make movies of those as well. And you can see these long, thin extensions that these glioblastoma cells extend into this brain-like tissue, enabling them to explore their local environment and potentially generate pulling forces to help pull themselves through that environment. And I'll show you more evidence of that later. And you can also see cells dividing. There's a cell that right here crawls up, divides, boom, now there's 2 cells. And so we can see the whole processes of migration and proliferation playing out in these brain organoid structures as well. And we can recapitulate this even in a 2D assay, which is a more traditional type of in vitro assay. But here we've engineered the substrate to be a hydrogel that has a Young's modulus that's more closely matched mechanically to what the Young's modulus of brain tissue is, more in the 5 to 10 kilopascal range. So, so these cells also have these long, thin extensions that reach out into their environment, enable them to get grip in their environment and pull themselves through it. And so this kind of extension retraction of protrusions is a key process that we'd like to understand in order to disrupt the invasive nature of these cancer cells. And so I'll tell you a little bit about the work that my former student Clarence Chan did when he was in my lab, which is developing a mathematical model for this process of those protrusions, which are driven by F-actin self-assembly at the leading edge to push the membrane forward in a ratcheting type mechanism to advance the leading edge and cause the cell to move in that direction. But confounding that movement at the same time are myosin 2 molecular motors depicted on the left here that pull on those actin filaments and pull them retrogradely back into the middle of the cell away from the leading edge. And then the third factor are these adhesive bonds, so-called molecular clutches that, like integrins and things like that, that can create mechanical linkages between the F-actin and the extracellular environment. And that they transmit force as the F-actin moves to the left under the influence of the motors and engage with the substrate. They can get stretched to varying degrees and start to transmit mechanical forces to a compliant extracellular matrix or extracellular environment. And so we have equations for those processes for both the motors and for the clutches. Whoops, sorry, jumped ahead there. For the motors and the clutches, which is that the motors have a force-velocity relationship. This should be fairly familiar from just going to the gym and lifting weights. If you go to the gym, you start lifting weights, you pick up a light weight, it's very easy. You can lift it about as quickly as you can contract your arm muscles. So there's like almost no resistance there. But as you increase the weight that you're lifting, eventually you get to the weight that you can barely lift or can't lift at all. That's the stall force of your muscle. And this is well known for ensembles of motors like skeletal muscle, but also true even down to single molecule level measurements of force velocity for single molecules of myosin and other motors like kinesin and dynein. So it's a well-established principle for motors. It's just true of motors generally. The more load you put on them, the faster they go. So the second part is what's the deal with the clutches? They engage and disengage spontaneously, but once they're engaged, they can start to come under load, which can then potentiate their off rate because they're being stretched. And that's kind of like pulling the bond apart. So there's an off rate that becomes force dependent, so-called Bell's law. It's well established going back to the 1970s that we also employ in this model. And with that all together, and then allowing stochastic bond formation and breaking in a load dependent way, we can simulate the formation and breaking of the clutch bonds, which are these small springs that you see in the middle here, transmitting force to the underlying substrate, which is this big spring. All the springs are color-coded according to force. So if it's blue, it's under no force. If it's red, it's under a lot of force. And you notice how they kind of warm up? Notice how the big spring, the substrate, loads up from blue to green to red and repeats this cycle and over and over. It's because this adhesion here that's formed with this ensemble of clutches is transmitting force from the myosin motors on the left that are moving the F-actin conveyor belt to the left. That's transmitting force to the underlying substrate and stretching it, and that force increases until it gets so great that the bonds are forced to fail because the loads on them are so great. And once a few fail, the rest start to fail in like a cascade domino effect. And that's a basic feature of the motor clutch model. So you could think about how, if this is the mechanism that glioma cells use to invade into brain, you might ask, okay, so what is the clutch that they're using? What's the adhesion molecule? Perhaps we could target it and block this process, and then they wouldn't be able to get grip on their environment, and they wouldn't be able to migrate. So we've tried to address that question, which I'll tell you a little bit about today, was really largely the work of Becky Klank-Markovitz when she was in my lab. And Becky did these computational models that built off the earlier model that Clarence developed, where instead of having just one protrusion, she'd have 3, 4, or 5 protrusions all kind of linked together mechanically in the middle, like the middle of the cell, so that all those protrusions are pulling on each other at the same time, all trying to go in different directions. And then it becomes a tug of war and a force balance, and eventually one of them wins out. And that causes the cell to displace itself in the direction away from the losing side. And you can see in these 3 different scenarios, cells move either well, like in the middle, or poorly, like on the left or the right. And what we've changed in the 3 cases is just simply the number of clutches that are in each of these simulated cells. So that on the left, there's very few clutches, which means that these protrusions get sent out and the myosin pulls on them, and there's almost no resistance to pulling them back in because there's very few adhesion bonds there. The few that are there break right away under load because there's so few of them, and they can't share the load effectively, and they get pulled back in. So they get these kind of retracted cells that aren't very motile. On the right is the opposite scenario, too much adhesion. So many adhesions that these protrusions go out and they basically get stuck. And the myosin can't overwhelm all those adhesions, and they pull like crazy, but they don't actually retract anything, and the cell just gets stuck like it's in mud. It also doesn't move well. In between is kind of a good balance between just enough grip to move, but not so much that you can't move on, which this cell moves pretty nicely off to the right within its frame. So that predicts that there's a biphasic speed dependence as a function of how adhesive the environment is. If it's too adhesive or not adhesive enough, cells won't migrate well. But if it's in between, they'll migrate really well. So that's a prediction we would make that we want to test experimentally ideally in in vivo or disease-relevant setting. It's been previously established in vitro using a variety of cells. Doug Laufenberger's work actually was very seminal in this area in the 1990s, but to our knowledge hadn't been applied in an in vivo setting in a disease-relevant situation. So that's what we undertook to do with working with John Olfest to investigate CD44 as a potential clutch in glioblastoma. That the idea being that CD44 has the right properties in that it spans across the membrane, has the ability to bind to F actin on the inside of the cell via adapter proteins like ezrin radixin or moisin, and can bind to hyaluronic acid on the extracellular matrix side, where hyaluronic acid is relatively abundant in the brain relative to other ECMs like collagen and fibronectin, which are not so abundant in brain. So working with John, who unfortunately passed away from metastatic melanoma at age 35, which was terrible, and Jim McCarthy, who's a CD44 expert, and John's student, Stacy Decker-Grunke, using a mouse model that John and Dave Largaspada had developed using the Sleeping Beauty transposase system, introducing oncogenic drivers in brown into neonatal mouse brains by injection of a plasmid cocktail, and using Sleeping Beauty and that can recognize, this transposase can recognize these black triangles, inverted repeat, direct repeat sequences in the transgenes as well as the host genome, to insert these transgenes into the host genome and fairly reliably drive the gliomagenesis in these mice. And you can pick oncogenic drivers that correspond to the human glioblastoma genetics, and so it has that advantage. It's immunocompetent animals, so you have the immune system in play too. And you can use pretty much any mouse you want, whether it's wild type or mutant. So by using a mutant mouse of CD44, we can start to control the CD44 gene dosage in the system to go to use knockout background to get to essentially no CD44, intermediate level of CD44 in the wild type case, or start to overexpress with a very active promoter, overexpress CD44 in both of these two backgrounds to get high or very high levels of CD44. And what we would predict based on our biophysical model is that the intermediate level of CD44 should be the fastest migrating, most invasive phenotype because they have the right balance of motors and clutches. And if you go too high or too low, they'll migrate poorly and the mice will do better. So that was our prediction, and then we tested that using, I showed you the movie earlier. You do this experiment for each of the 4 cohorts to see what the speeds look like and what the survival looks like. And what we found was exactly what Becky's model predicted, based on the motor clutch model, that the wild type intermediate level of CD44 had the fastest cells and the poorest mouse survival. And so you see this biphasic curve, both for migration and then upside down survival, the corresponding inverse of that poor survival at intermediate level of CD44, which prompted us to look at human CD44 mRNA expression to see if they similarly exhibited this kind of biphasic trend. And indeed found that was the case in this cohort. And so that intermediate level of CD44 correlated with worse outcomes relative to somewhat better outcomes at either low or high levels of CD44. So to kind of summarize that whole story is basically that we had evidence that CD44 has the right properties to be a clutch for glioblastoma cells in their invasion into the brain tissue, such that there's just the right balance of motors and clutches, of myosin motors and CD44 clutches. So we've done a lot of other studies on the motor clutch model, and other groups have used our model too to make predictions that led to new discoveries. And I focused on the migration of brain tissue just now, but we've also used it to discover negative and other aspects of cell biomechanics and migration in complex environments. So what I'd like to tell you about next is, can we find direct evidence that they're actually using this motor clutch mechanism in brain tissue? And this is the work of my former student, Sarah Anderson, who made movies of cells migrating through brain tissue and considered a competing model to our model called the osmotic engine model, which was developed by Konstantin Konstantopoulos, Sean Sun, and Denny Wirtz at Johns Hopkins. The idea of this model, which is quite interesting, I think, is that there are electroosmotic pumps at one end of the cell that drive ions in, which then in turn draws water into that side of the cell, which pressurizes the cell and causes the water to flow out on the other end of the cell. And due to that imbalance of electroosmotic pumping, you get a net flux of water through the cells. And by conservation of momentum and mass, the cell moves to the right along with that. It doesn't need to touch anything. It just like cruise through the environment. As long as it has that asymmetry, although it's helped by having a seal along the sides to maintain the asymmetry. So it's a plausible biophysical model, in my opinion. We wanted to take it seriously because they had proposed that glioblastoma cells might actually use this mechanism to invade through brain. So taking their model seriously, considering what the predictions would be between the two models, the motor clutch model on the left, as I mentioned, predicts these pulling forces on the environment. They're pulling in on the environment as they grab it. So if you were to sit and watch a cell as it's moving, it's going to pull on the environment, deform. If it's sitting next to like a blood vessel, it's going to grab onto it and pull it and deform it. And you should see the deformation pulling towards it. And as one side fails, just because that's what happens when the forces build up, one side eventually loses. Then the cell will move in the opposite direction. And so you'll see at the leading edge of the cell, the vasculature would move towards the cell as it invaded in that direction of the vasculature. That is in contrast to the prediction of the osmotic engine model, which predicts that as these fluxes pass through the cell, as the water fluxes through the cell moves autonomously, and any adhesions that get made would be basically resisting that motion and would get dragged along with the cell as it moved to the right in this scenario. So that would predict that if you could visualize the deformations in the vasculature, they would move along with the direction of the cancer cell as it moved to the right, an opposite prediction. So one model predicts that they move towards each other. The other one predicts that they move together in the same direction. So Sarah tested this experimentally by fluorescently tagging the vasculature using isolectin B4, which also happens to tag the microglia in the environment, and green fluorescently tagging the glioblastoma cells, and observing as they migrated in a perivascular manner, this cell moves down and to the right along this stretch of vasculature indicated by the magenta arrow. And what she predicts is that it would pull it towards it as it moves to the right if it's the motor clutch model, which a little light in here, but maybe you can see it. You can see that stretch of vasculature just get sucked in, pulled in by the glioma cells. It moves down and to the right. You can make a chymograph in that region there and track the features of magenta and green, and they exhibit this kind of convergence that you'd expect from the motor clutch model, and they tend not to exhibit this kind of pushing dynamics that the osmotic engine model would predict. So you see this kind of V shape in the chymograph of the green and the magenta coming together. And she did this for a number of different cell lines too, including ones from the Mayo PDX collection. So we're grateful to Jan and his team for providing us with the cells to examine in the brain tissue. And the same features consistently across all these different lines see this kind of pulling behavior. You can see the magenta getting pulled down to the left in the rightmost movie. So Sarah went through and scored all those events to see if there's examples of pushing or pulling. And pretty much almost every single time, but not totally, with one exception. She saw one example of what looked like a pushing event here, but otherwise all the other events of deformation that were associated with movement were these pulling type events that I just showed you examples of. So to us, this is direct evidence of what you'd expect from a pulling type mechanism without evidence to support evidence of a pushing or osmotic engine type mechanism. And yeah, I'm going to skip this real quick. She showed that she could block CD44, and that suppressed the ability of the cells to move in the brain tissue. And kind of interestingly, didn't really adversely affect the T cell migration, which I probably won't get to those movies today, next time. But it actually potentiated slightly the T cell migration in brain tissue. So overall, just to wrap up this first part here, the glioma cells seem to use a motor clutch mechanism in brain tissue that is CD44, and I didn't mention it, but also integrin and myosin II dependent. And they don't seem to exhibit behavior that's consistent with the osmotic engine model. So we disfavor the osmotic engine model and favor the motor clutch model for invasion, and CD44 playing an important role in that. So then we started to think, well, how can we target glioblastoma cell migration? Can our model predict what the best things to target are and what the not so good things to target are? And so my former postdoc, Jay Ho, who's now assistant professor at Brown University, started to simulate what would happen if you changed any one of the parameters in the model to see how that would affect how fast the cells migrate. And that's shown here is that this cell migration simulator that I showed you earlier, when you change any one of the parameters shown here in the rightmost column, you see an effect on the random motility coefficient or speed of the cell. But the biggest impact on that was largely the F-actin assembly dynamics, V-poly. If you increase that, you can increase the speed a lot. And so that's shown here as the RMC in the top row here. As you go from low, medium to high actin polymerization rate, goes from relatively low speed to relatively high speed. And so that made us think, well, rather than targeting the clutches, maybe we should target the F-actin assembly. And so, or really, you could target any part of this potentially and have an impact negatively if you target it the right way on the random motility of the cells. But the standout champ was the actin dynamics. So my current student, Katie Vopat, started to think about ways to target F-actin dynamics. And we're particularly struck by the feature of these spiky projections that glioma cells send out into the brain tissue, which are very much like reminiscent of so-called philopodia, which are parallel bundles of F-actin that are cross-linked together, I think I have that in the next slide, by bundling proteins such as this protein fashion, which is the yellow circle here, bundles these together into a nice bundle and keeps it together so it has a nice spiky, pointy protrusion, and those things can poke into the brain tissue and help the cells generate protrusions that go through it and help them navigate through the brain tissue. So she was intrigued when she found a paper that said that an antidepressant called imipramine can actually block fashion 1 interaction with F-actin and can block this bundling process. And so we were intrigued because if that's true, then it might be able to block or suppress glioblastoma cell migration and invasion. And here we'd have a drug that's already used clinically. It's brain penetrant because it's an antidepressant. So we thought this would be an interesting angle to pursue potentially. So Katie started to look at imipramine as an anti-migratory drug and found that, maybe let's restart this here. These cells initially have these long spiky protrusions, which when she has imipramine on the left versus control on the right, those rapidly disappear. So you lose those spiky protrusions. The cells kind of retract and round up, and they become relatively non-migratory in the presence of imipramine. And this is very preliminary and early. That's why I hesitated to show it. But I thought, let's just take the chance and roll the dice here and talk a little bit about a possible drug that we could use. And this is a very inexpensive drug. It costs $10. It would cost about $10 for a course of treatment. I looked it up on Mark Cuban's gooddrugs.com. So, and still early days, but so far it looks like a statistically significant decrease in cell speed at even like 10 micromolar imipramine. So we've got more work to do, obviously, but we've tried to test it in vitro on hyaluronic acid gels to be more closely mimicking brain tissue. And we've tested in the brain slices too. You're not seeing the red vasculature too well here, but you can see the green glioblastoma cell when it's treated with imipramine. I'll start it over again. You see the long, I can't see it that great, but these long protrusions here at imipramine, and they basically go away. And the cell becomes non-motile, kind of stuck right here for an extended period of time, for several hours. So it's showing evidence of anti-migratory activity in brain tissue as well as in vitro. So we think it might be an intriguing drug to try as a anti-migratory, anti-invasive drug for glioblastoma. There's actually evidence that it is anti-invasive in glioblastoma based on earlier work by Jenny Munson when she was a PhD student in Ravi Bellamkonda's lab at Georgia Tech, and found that, as you can see here in the imipramine-treated case, a decreased number of invasive cells in this rat model for glioblastoma, and evidenced here as well in this histology with quantification here in the top row. And they tried to work out the mechanism, but it was a little vague in their final interpretation. They thought it was targeting this NADPH oxidase NOX4, and eventually, maybe perhaps signaling through SARC, and eventually affecting actin. So I guess what I'd say about all this is a lot of question marks here, so not really completely worked out how it works as an anti-invasive, but it had that effect in the mouse, in this rat model for glioblastoma, and it seems to end up involving actin again. So that's consistent with the article I mentioned that Katie found of the group that found that imipramine blocks fashion, the F-actin bundler. It has this actin-directed effect that can affect invasion. So can we do an imipramine clinical trial for glioblastoma? I'm currently discussing with our clinical team at Minnesota, trying to get a protocol together to do this. The two clinicians that I talked to, one is treats human patients. The other one treats canine patients. So we have one of the top canine clinics in the United States, formerly by Liz Pluhar, now directed by Dr. Susan Arnold. And Dr. Arnold's comfortable with trying to use imipramine with the dogs who have spontaneous gliomas. They're not experimental animals. They're pets that need treatment. And she uses it already in the clinic for dogs. It's already used clinically for dogs to treat separation anxiety and things like that, and urinary control. So she's comfortable with exploring the idea. And I appreciate getting feedback from this group. If you think this is a terrible idea or there's reasons why it's not going to work, now's the time to tell me, because I think we're going to kind of keep pursuing this as long as the preclinical data keeps supporting it and the clinicians want to go along for the ride and feel like it could help their patients and not harm them. I'll just mention, too, it could have pleiotropic effects, and this has been investigated by Doug Hanahan's group in Lausanne, which is that it seems to have other effects on the immune system, including macrophage reprogramming and inducing recruitment of CD4 and CD8 positive T cells that are beneficial to suppressing tumor growth. So it doesn't rule out these other pathways, how that works, they didn't fully work that out either, although they suggest autophagy as a mechanism by which that happens. But when you start interfering with actin dynamics, all kinds of things can break loose in the cell that may manifest as autophagy or signaling alterations and things like that. So there could be pleiotropic effects. And there are clinical trials open to test imipramine for glioblastoma. One is in Texas at UT San Antonio, MD Anderson branch in San Antonio, and testing it in recurrent glioblastoma. And another one is in Spain, which is motivated by the group that discovered that imipramine blocks fascian activity in Cartagena. And so they're enrolling triple-negative breast cancer patients and colorectal cancer patients and treating them with imipramine and assessing their outcomes. So it's already happening. It's not like we invented this or came up with this idea. What we're excited about is the modeling that we're doing pointed towards F-actin assembly dynamics as a key thing to target. And thanks to the group in Cartagena discovering that imipramine targets fascian and blocks bundling, we seem to be backing it up with our preclinical data. And so that's why we think it all kind of makes sense mechanistically to go after glioblastoma with imipramine as an anti-invasive strategy. So I have my whole immune cell migration stuff, but I think I've used up all my time and then some, and Forrest is agreeing. So but at least he's not reading the newspaper. So I'll stop there, and if there's time for questions, I'll be happy to answer them. And if you want to give me feedback on this idea, that'd be great. If you think it's terrible, let me know. If you think it's great, join us, cheer us on, join the conversation. Yeah. Thanks.
Questions?
You said it was 40 micromolar that you were using of the imipramine to see the effects in vitro. Do you know much about the pharmacokinetics of it and what concentrations you get in the brain and so forth?
Yeah, I mean, we've started to look into the literature on that. So it seems like 1 micromolar might be a reasonable, attainable concentration that's used for treating depression already. So, and we've taken it down to 1 micromolar. I didn't show the data, but I mean, that's where we're headed. Is there was no drop off. I mean, the reduction in RMC was already fully evident at 10 micromolar and didn't get any, there was no more suppression of migration when you went to 40. So it's already saturated out at 10. And Katie's seeing suppression at 1 micromolar in vitro as well. So we think we can see effects at 1 micromolar, and we think that looks to be a clinically achievable concentration.
David, what happens to the tumor cells after treatment with imipramine in terms of, you say they don't invade, right? You block invasion, but now they stay there, right, in the original location. Do they proliferate more? Do they proliferate less? Do they die? I mean, I think answering this question is quite important. For example, I would test their response to standard of care, right, to radiotherapy and chemotherapy. Are they becoming more sensitive, less sensitive? Because, you know, obviously invasion is important, but you also want to make sure that you don't make cells more aggressive for whatever reason.
Yeah, I mean, we don't have those results yet, but the other groups that I mentioned, Manson and Balamconda, and then the group in Spain, they've seen that it has beneficial effects in the animal models that they've looked at. But you've raised an important question, I think, is like, so what if you stop them from moving? They keep proliferating. Right. But I would argue they proliferate in place now instead of like moving out and proliferating. And I think there's this problem of, I mean, one thing is what that does is it creates a very compact tumor that's jammed together, which makes it hard for cells to proliferate that are in the tumor. Because why? Because there's no space to put the daughter cells. So expanding, for the cell to expand to get to twice the size that it was initially as it goes through the cell cycle, and we've been discussing over the last year or so with Scott Manalis's group the biomechanics of cells actually growing and the physics of that. You got to have space to grow. So it's not a direct effect on proliferation, but you can't proliferate if you don't have space to grow into. And so that's one thing. Two, as I mentioned, is like the immune system has this big problem with diffuse tumors. When they're that sparse and spread out, you got to skip from one to the other to be a serial killer. You might spend like hours trying to find the next cancer cell. Meanwhile, that cell's growing, as are all the other cancer cells. So I think it's going to make the tumor more vulnerable to immune therapy and normal immune response if we can keep it in place more. So those are two reasons why I think anti-migratory therapy is still exciting, even if it doesn't directly stop migration. I mean proliferation, sorry, Antonio.
Great talk. Yeah, I was just wondering, thinking about your the implications of your model in this immunotherapy context, would delivering an agent like imipramine, could it at all interfere or become antagonistic with lymphocyte migration throughout the tumor, or perhaps have some on-target toxicity with the normal brain?
Yeah, that's a great question, and it would have been touched on if I'd done the third half of my talk, but I'm not going to do that, so I'll just try to give you the short answer. When people look at T cells migrating and lymphocytes kind of generally, they tend not to have these really spiky protrusions like you saw with the glioblastoma cells. They oftentimes have this more amoeboid or rounded bleb-like leading edge in their migratory mode. And we've investigated that scenario now using a biophysical model for blebbing too, and amoeboid migration. And what we've concluded is that cells should be able to move just fine and at very fast speeds if they use blebbing combined with adhesion. But blebbing dynamics alone without adhesion is ridiculously slow and can't explain what you see with T cells and brain tissue. I could probably just give you I'll just show you a movie just to get you thinking about it. Here's T cells migrating in brain tissue. Same assay as before, but now we swapped in T cells instead of glioblastoma cells in green. And again, magenta is vasculature and microglia. And what you see is a lot of green cells that aren't moving, right? They're just sitting there. But the ones that can move very fast. Like watch this champ here, right there, back and forth, then out. And when they move really fast, like 10, 20 microns per minute. But a lot of cells aren't moving. So we're concerned about the therapeutic cells that come in there and just don't do anything. They just sit there. Time's ticking, you know. They got to go get killing. Don't sit there and wait. So we want to understand why they're waiting. We want to understand what limits their speed. But they don't seem to really use these filopodia so much. They can do that. They're capable. We've made like a whole, in vitro, we can see all kinds of protrusions of T cells, some of which look kind of spiky, like this guy here with that thing. So if you treat these cells with fashion, they've got other options to go explore the space that don't require filopodia and actin bundling to do it. So our prediction is that they won't be very sensitive to imipramine in a negative way. But in fact, we need to test it. So that's on the list there. Thanks.
So I had a quick question, Dave, about, that was an awesome talk, by the way.
Thanks.
But I had a quick question about sort of other cytoskeletal events that might happen, and thinking about sort of cells undergoing a mesenchymal transition, for instance, where you see a lot of cytoskeletal rearrangements. I don't know that those are driven by FASN, so maybe this drug wouldn't affect that, but have you guys thought about that or looked at that?
Yeah, I haven't thought too deeply about it. I mean, Katie got me excited about imipramine about like 2 months ago. So what? Imipramine? What is it?
So what's taking so long?
I don't know. Sorry, of course. I'll come back next week with a full report. Yeah, no, I haven't really thought about it too much, but certainly my thinking, once you change even just the shape of the cell, you've kind of changed everything.
Yeah.
Because you've changed the architecture of the cell, and that affects function. It's very hard to have that not affect signaling, to have it not affect other parts of the cytoskeleton, to have it not affect metabolism. It's almost unavoidable. That's why I think these pleiotropic effects probably kind of make sense, that it's actually mostly targeting fashion. I'm not sure about that, of course, but it could be multiply targeting a lot of things. But it seems more likely that it really targets fashion, and then the downstream impacts of that play out differently depending on which cell they hit, whether it's a macrophage or a T cell or a glioma cell.
Cool.
It'd be cool if all the ways it played out were all in favor of a better outcome in the glioblastoma.
So have you thought about like the mechanism of action of why the drug that you're focusing on, inhibits the mechanistic action? Because it's a tricyclic compound and there's so many like diazepines or things that are already FDA approved. Have those been looked at for migration inhibition, or is it something about this like moiety of this drug that specifically targets that, migration aspect?
I mean, we're following up on the fashion discovery there's a group in Cartagena, Spain that discovered this, and I cited their paper very briefly, which is this paper, which I didn't go into any detail about, but they actually did a docking study with these tricyclic compounds, including imipramine, and found that fashion was one of their top hits in a simulated, kind of docking attempt. So I guess that's kind of what. Now you might wonder, well, how does that explain its antidepressive activity? I'm not an expert on psychology or psychiatry, so I don't know. But what I might think, a reasonable hypothesis to me, is that if it's interfering with F-actin dynamics, of course the brain has its wiring is via F-actin protrusions to form dendritic spines and things like that. So if you interfere with that, you're going to interfere with the normal dynamics of brain architecture, and that may manifest as it's a reuptake inhibitor, which is how it's described in the literature typically. But at the mechanistic molecular level, might actually be through fashion, for all we know, until someone finds other target. Yeah.
Thanks.
Yep.
All right. If there's no other questions, let's thank our speakers for the day, and thank you all for attending our first inaugural, I guess that's redundant, but anyway, our inaugural symposium for our center. So hopefully we'll see you next year at our second non-inaugural symposium. Cool, thanks.